So for this DNA extraction experiment, I will be using two fruits, banana and kiwi. The purpose of this experiment is to help us understand that DNA is in the cells of all living organisms, including in the foods we eat. And to do this experiment, the materials we will need are dish soap, Ziploc, cold water, strainer, glasses, teaspoons, salt, knife, and cold isopropyl alcohol. So now the first step to do this is to pour cold water into the glass until the half. Then add 6 pumps of dish soap to the water, and this helps pull apart the fats and proteins that make up the membranes surrounding the cell and nucleus. Next, stir in half a teaspoon of salt and mix it until it fully dissolves. Salt is important because by adding salt, we help neutralize the DNA charge and make the molecules less hydrophilic, meaning it becomes less soluble in water. The salt also helps to remove proteins that are bound to the DNA and to keep the proteins dissolved in water. After that is done, we set that aside to now slice the banana and the kiwi. Sorry miss, I don't know how to slice a kiwi. Okay, so now once that is done, we now place the fruit onto their own Ziplocs. Then, we now pour 5 spoons of the extraction mixture into the bag in each fruit. Then make sure to remove as much air from the bag as possible and seal it closed. Make sure to mash the banana and kiwi inside properly until there are no large pieces remaining. Now once that is all done, pour the resulting banana and kiwi pulp and extraction mixture through a strainer and into a small glass. To make the procedure easier and faster, use a spoon to press the mashed bits against the strainer, forcing even more of the mixture into the container. Next, we add 2 teaspoons of the chilled isopropyl alcohol into the solution. Now this is important because DNA clumps are soluble, meaning it can be dissolved in some liquids but not in alcohol. So adding alcohol helps the clumps of DNA to form. And now we wait and observe for the results. Finally, use a fork to gently remove the DNA from the solution and lay it on a dish to further examine. So as you can see based on my results, it is very obvious that the banana had a more visible DNA than the kiwi. The reason for that is because the banana yields more DNA than other fruits, and second is because they are triploid, which means that each banana cell has 3 sets of DNA, so there is a lot of available for extraction. For the kiwi, it is seen that the DNA was not really visible, and I believe that it's because it is a much harder fruit to pulverize.